topic that we'll be dealing now is about the antibodies. So antibodies are major effector molecules of the humoral immune response. So immunity is the resistance against the different types of infectious agents. Apart from infectious agent, they also provide immunity against the bacterial products such as the toxins. So it is resistance against the different organism and the organism that produces certain toxic products. So resistance of a body against this organism is called as immunity. And immunity is of different types. It is either innate immune response or adaptive immune response, where both the immune reactions come forward to protect our body from the different types of pathogenic microorganisms. And coming to the adaptive immune response, also called as acquired immunity, which we acquire it during our lifetime. And this acquired immunity can be brought about either by the humoral response or by the effector response. So how it responds towards a particular organism can either be in an humoral branch or the cellular branch, but ultimately it is all the system that comes forward to protect our body against the different types of infections. So the humoral branch of immune response deals with the production of B cells that produces the antibodies. So the antibodies are a major effector function of the humoral branch, whereas the cellular immunity is usually provided by the T lymphocytes. Now depending upon where they are usually produced and measured, you give them the name as B lymphocytes or the T lymphocytes. That is in the bone marrow it is called as a B lymphocyte, and if it is in the thymus then it is called as the T lymphocyte. So we are majorly concerned with the antibodies that belongs to the humoral branch of immune response. So here the procedure or the steps that takes place to produce the antibody is, when an antigen enters into the body, it activates the immune system to produce the lymphocytes, either the B lymphocytes or the T lymphocyte, that carry specific markers to go and interact with the antigen specifically and in an observable manner. So it is in the sera that these antibodies usually reside. So the antibodies are nothing but the serum components that are produced they are produced when there is an antigenic interaction into the human body, and they react specifically. As told before, there are certain markers that are present on the B cells such as antibodies that goes and interacts with the T cells, and it is highly specific because it is based upon the complementary shapes between the antigen and the antibodies. And the presence of antibodies in the immune following a chronic infection is called as the immune sera. Now the presence of these immune cells, that is the B lymphocyte in the sera, was first demonstrated by Tiselius, where the experiment was carried out to separate the serum component based upon the chemicals that are used, that is ammonium sulfate. Fractionating with the ammonium sulfate, you can classify the human sera or separate the human sera into two components. One is soluble albumin and the insoluble globulin. So here the insoluble globulin can be again further separated into the water soluble pseudoglobulin and euglobulin. So it is in the euglobulin that you can see the antibody activity. Later on, Tiselius and Capet has electrophorosed this albumin component in an electrophoretic mobility to separate them into albumin, alpha, beta and gamma globulin. So ultimately it is a gamma globulin that contains the antibody activity. So antibody resides in the serum in the gamma globulin portion, which reacts with the antigen whenever there is an interaction of antigen into the human body. And now there are different types of antibody molecule with different functions. So in order to overcome the problem of giving them a name depending upon their functions, finally a name was given which are called as immunoglobulins. Now these immunoglobulins are the one that are nothing but which contains the antibodies which carry out the humoral response of the immune system. So immunoglobulin is a broad concept, whereas the antibodies are the one that usually carries out the major effector functions of the humoral branch. So immunoglobulins are structural and chemical concept, you can say, whereas the antibody are the one that carries out the biological and the functional properties of the immunoglobulin. Now there are 20 to 25% of the total serum proteins. They are weight about 20 to 25% of the total serum proteins, and all immunoglobulins, all antibodies can be immunoglobulins, whereas all immunoglobulins need not be antibodies. Now these antibodies are based upon the physico-chemical nature and the type of function they usually carry out, you can classify them into 5 major type of antibodies. They are IgG, IgA, IgM, IgD and IgE. Later on we will study about the different important properties of this particular classes of antibodies. Now coming to the structure of the antibodies. So how is the structure and how it goes and interacts with the antigen is very important for us to understand the specificity between the antigen and antibody. So it was Potter and Edelman who deduced the structure of antibodies by 3 important steps. One, they have carried out with the enzymatic digestions with the papain and the pepsin, and finally digested with the chemical to know the complete structure of the antibodies. So when the IgG molecule was first fragmented or digested with the papain, you get 2 fragments. One is called as the FC fragment, the 2 fragments are called as the FAB fragment. FC fragment is so called because it crystallizes while during the cold storage. That's why the name was given as FC fragment. So 1 fragment of FC was produced and 2 fragments of FAB was produced. FAB is nothing but antigen binding region of the antibody molecule. Now, this antigen binding region of the antibody is very important for the antibody to go and interact specifically with a particular type of antigen. So when digested with the papain, they produce 2 fragments. One is the FC fragment, which crystallizes during the cold storage and hence it is called as FC, and the other one is called as the FAB fragment, antigen finding or antigen binding portion of the antibody molecule. Similarly, when the IgG molecule was digested with pepsin, they could see only the FAB fragment, 2 types of FAB fragments, whereas FC fragment was not produced because it was completely digested by the pepsin. So the FAB fragment is the one that interacts with the antigen molecule, and ultimately when this IgG molecule was treated with mercaptoethanol, they could see that these FAB and FC portions are usually joined together by the multiple disulfide linkages that helps the complete antibody molecule. So it is how they deduced the complete structure of the antibody molecule, having 2 important portions. One is the FAB portion and the FC portion. FAB is antigen binding portion. Depending upon a particular type of antigen, this type of antibodies are produced, and the FC portion is crystallizable portion, which is not involved in the antigen binding, but it carries out various biological effector functions of the humoral response. So here we have the structure. One is the FAB portion and the FC portion. Now these antibodies are said to be glycoprotein in nature. Glycoprotein means they are made up of both protein as well as carbohydrate. Glyco means carbohydrate and protein are made up of the amino acid. So here we have different type of polypeptide chain. So as it is a protein, it will have an amino terminus and a carboxy terminus. So there are 2 pairs of such polypeptide chain that are held together by disulfide linkages, and it is a complete structure of antibody that is of 2 pairs of polypeptide chain. Now each antibody molecule has 2 parts. One is called as a light chain and the other one is called as the heavy chain. Now coming to the light chain has a molecular weight of about 25,000 daltons, and it is held with the heavy chain by disulfide linkages, and each light chain is made up of either a kappa particle or a lambda particle. So named after the discovery by the scientists, kappa means corn gold, and Lapari had discovered the lambda portion of the light chain. So it is either the kappa chain or the lambda chain that can be present in an antibody molecule, but not both the kappa or the lambda. It is either kappa that can be present or the lambda, and the ratio of this K to L chain, lambda chain in our human body is 2:1. So remember that it is either kappa or lambda that can be present in the heavy light chain, but not both the component that can be present in the light chain. Now next, coming to the heavy chain, it is genetically and biologically distinct and carries out different function, and it weighs about 50,000 daltons and structurally and antigenically distinct, and based upon that you can classify the antibodies into 5 major types and given the name on the Greek letters, that is IgG, IgA, alpha, IgM, mu, IgD, delta, IgE, epsilon. So they are usually named after the Greek letters, and they carry out different biological functions in the human body. Now coming to the variable and the constant region. So each light chain and heavy chain are divided divided into two major components, variable region and the constant region. The variable regions are nothing but the regions that are present at the amino terminus, and here the amino acid sequence varies from one antibody to the other antibody, and it is this region that specifies the specificity with the different types of antigen. It changes with the different types of antigen. That is why it is called as variable region. So among the 214 amino acids that makes up the light chain, 110 amino acid in both heavy chain and the light chain are highly variable, and they are the one that usually goes and interacts with the antigen. So they are the one that are the portion of antigen binding portion. And then we have the next followed by the constant region, which is constant in all the type of antibodies. So there is two major classes. The variable region present in both heavy chain and light chain, that is designated as VL and VH, antigen binding region, and then we have CL and CH, the constant region present in the light chain and the constant region present in the heavy chain. And it is in the variable region, as I told you, that is involved in the antigen binding region. It is not all 110 amino acids that are involved in binding with the antigen, but there are certain hot spots which are called as hypervariable region. Few amino acids in 110 amino acids are the one that interacts with the antigen, so they are called as hypervariable region or the hot spots. So among this hypervariable region and hot spot, there are certain sequences of the amino acid that directly interacts with the antigen, which are called as complementarity determining region. As already discussed, there is a complementarity between the shapes of antigen and antibody. So this complementarity determining regions are the one that ultimately goes and interacts with the antigen that has entered the human body. So it is the variable region. In the variable region, it is a hypervariable region, and within the hypervariable region, it is the complementarity determining region that goes and interacts with the antigen. So in the light chain, there is equal amount of the variable and the constant region, whereas in the heavy chain, 1/5 of it is the variable region, the other part is the constant region. And the FC portion that is already discussed, it is a crystallizable fragment of the antibody molecule. Though it is not involved in binding with the antigen, it induces different biological activities in the human body to eliminate the antigen. The biological activities that are brought about by the FC portion is they can activate the complement system, they can enhance the phagocytosis, which is called as opsonization, they can mediate the antibody dependent cell mediated cytotoxicity. Apart from that, they can activate the other complement system also during the complement activating system. So here the FC portion is involved in the biological effector function, whereas the FAB portion is the one that interacts with the antigen. And these the light chain and the heavy chain is usually attached to each other with the help of a disulfide linkages. So this is called as the interchain disulfide linkages. Similar type of disulfide linkages are present within this light chain and the heavy chain. They are usually forms a loop, and they have a globular domain, and they have their own property to interact with the antigen. So they are interchain disulfide linkages and intrachain disulfide linkages that helps the complete antibody molecule. And apart from that, the CH1, CH2, CH3 and CH4, these are the constant regions that are present in the heavy chain. They are usually not seen in the light chain. Only one portion of the light chain has this constant region, whereas 2 to 3 portions can be present in the heavy chain. So this CH2 and CH3 are mostly involved in the activating the component protein or the complement protein, and then CH3 is usual in the adherence of the mononucleoids with the like bacterial surfaces, and it is between this CH1 and CH2 that there is a hinge region that usually connects the FAB portion and the FC portion. So a hinge region is a flexible portion that helps to make the antibody move depending upon the base pairing between the antigen and the antibody. So hinge region is very important to bring a complementarity shape to get attached to the antigen that is usually interacting with the antibody. And here we have different classes of antibodies. As already discussed, they are for different physico-chemical nature, and they are structurally and genetically different in present in the constant region of the immunoglobulin molecule. So there are 5 different types of or the classes of antibody based upon their function. They are usually given the name as IgG, IgM, IgD, IgE and IgA. So first I will be dealing about the IgE, and the concentration of this IgE is very high in the human serum. There is about 80% of this IgG that is present in the human serum, and it is the you can say it is the most prominent one and the most functional one to interact with the antigen that has entered via the blood or the tissue spaces. So the first you can say the antibody that interacts with the antigen, and it has a molecular weight of about 150,000 Daltons and a half-life of about only 23 days. And it is the only antibody that has the ability to cross the placenta and provide immunity to the fetus who is not able to produce his own immune response. So during the pregnancy, it is the antibody that goes crosses the placenta and reaches the fetus and protects the fetus against variety of infections such as rubella, intrauterine infections caused by different types of diseases. So has the ability to transport across the placenta, and they can are also involved in the phagocytosis, that is called as antibody dependent cell mediated cytotoxicity, and these antibody which enhance the phagocytosis are called as the opsonins. And when this IgG molecule reacts with the platelet Fc receptor, they help to release vasoactive amine that usually brings about a biological effect in eliminating the organism. And apart from that, they also participate in various immunological reactions, and it is said to be a general purpose antibody because it interacts with the antigen or the organism that enters via the blood or the tissue spaces. And this is the antibody that is mostly given as an isoimmunization or anti-RHD IgG for lady to overcome this disorder which is called as erythroblastosis fetalis. So immediately after the delivery, this type of antibodies are given to suppress the production of antibodies that kills the baby in the next pregnancy. So that is called as erythroblastosis fetalis. So isoimmunization with this IgG antibody prevents this erythroblastosis fetalis in a pregnant lady. And there are variety of classes of IgG antibodies. They are IgG1, IgG2, IgG3 and IgG4. The concentration of IgG1 is 65%, IgG2 is 23%, IgG3 is 8% and IgG4 is 4%. So this is the IgG antibody that brings about the major effector function of the humoral immune response. Next, the next type of antibody is called as the IgA, immunoglobulin alpha. That is the second most abundant that can be seen in the human serum, and it is about 10 to 13% of the total serum proteins. And this IgA antibody is called as the secretory antibody because it is mostly seen in the secretory fluid such as the colostrum. It is seen in the tears, and they can also be seen in the sweat. So the normal serum level of this IgA antibody is around 0.6 to 4.2 mg per ml, and the half-life is around 6 to 8 days, and secretory because it is present in the colostrum, the first form milk, and it is seen in the saliva and the tears. And there are two forms of this IgA antibody that can be seen in the serum. One is called as the serum IgA, which is monomeric in nature, and the second part is that is produced by the plasma cells near the mucosal or the glandular epithelium cells, which is called as a secretory IgA. It is a dimeric form which is joined together by a chain which is called as a J chain, and the J chain is called as a joining chain. Just join the two antibodies together. So IgA is a dimer, and it can also be in a monomeric form. So it is called as a secretory IgA, produced by the plasma cells present near the mucosal surfaces and the secretion, and joined together by the J chain. And apart from the main dimer, it also has an extra component which is called as a secretory component produced by the mucosal or the glandular cell. It is not produced by the plasma cell, but it is produced by the mucosal or the glandular cell, and this protects the IgA from the digestion by the bacteriolytic enzyme that are produced by the bacteria that are present in the human intestine. So it protects the IgA by getting cleaved with the help of bacteriolytic enzyme. So this is called as a secretory component that is attached to the normal IgA molecule, and it can transport it across the luminal surface because of the presence of the receptor, that is a secretory component that is attached to the IgA molecule, and it is the receptor that helps the IgA to get transported across the luminal surfaces. And this antibody molecule usually forms an antibody paste. So they usually attack the antigen. They form a paste around the antigen and will not allow the antigen to go and bind to the mucosal surface, thereby preventing their entry into the tissue spaces. So they provide a local immunity where they will not allow the antigen to go and bind to the surfaces and get gain an entry into the tissue spaces. And they are resistant to digestive enzymes because of the presence of a secretory component, and they inhibit the adherence of microorganisms by forming antibody paste, and they are the one that can activate the alternative pathway of the complement system and phagocytosis. And they do not activate the complement system directly, but only pathway that can be activated by IgA is the alternative pathway, and they also helps in enhancing the phagocytosis, acting as opsonins. And there are two subclasses of IgA. They are called as IgA1 and IgA2. So next, coming to the third type of the antibody molecule, the class of antibody molecule, it is the IgM. IgM is called as a millionaire molecule because it has 5 different types of antibody molecule that are linked again with the J chain. So it constitutes about 5 to 8% of the total serum component. That is, the antibody present is about 5 to 8 in a total serum component, and the level is around 0.5 to 2 mg per ml, and it has half-life of only 5 days. So it represents that the half-life is only for 5 days. So the presence of the IgM antibody in the serum indicates a recent infection in the human body, and it is called as a millionaire component because it is made up of 900,000 to 1,00,000 daltons, and it is a pentamer having 5 antibodies joined together by a J chain, and it can be seen only in intravascular component, whereas the IgG molecule can be seen both extravascular and intravascular. This IgM is only present intravascularly, and it is the earliest immunoglobulin that is produced in the fetus after the fetus develops his immune system. So the presence of IgM in the fetus helps us to identify the organisms or the infections that the fetus or the newborn child is usually having, and this IgM can be used in the diagnosis of congenital infection in the fetus or the newborn baby that has developed his immune system, and also helps us to identify the rubella or HIV infection and toxoplasmosis. And it is a short-lived, as told, shows about the recent infection, and it is a isohemagglutinin, that is the person with the A blood group will have B antibodies, so they are called as anti-A antibodies. So the antibodies present are usually produced again with the cross reaction with the intestinal flora. So these are the antibodies that are of IgM type. So apart from that, the antibodies are also active against the typhoid O antigen and reagent antibody, and it has a unique structural feature, and the antibody IgM can be expressed if it is in a monomeric form. It is usually expressed on the surface to helps in activating the B cell for proliferation and differentiation, and the deficiency of IgM can lead to septicemia, where there is a generalized infection with the different types of microorganisms. So it prevents the generalized infections by the pathogenic microorganism, and is a major antibody receptor that can be seen on the B cell apart from the IgD. So IgD and IgM are the one that are usually expressed on the B cell, which helps in the clonal selection and helps in the proliferation and differentiation of the B cell. Next, the important class of antibody molecule is the IgD. It is very less amount of IgD that can be seen, and very few biological properties are contributed by this IgD. It is only about 3 mg per ml, and half-life is about only 3 days, and they also act as a recognition receptors on the B cell, similar to that of the monomeric IgM that was expressed on the B cell, and it helps to either proliferate a B cell or suppress a B cell depending upon the antigenic selection, whether the B cell has to undergo proliferation or it has to go suppression. So that is what is called a clonal selection. So when there is no requirement of any antibodies, the B cells are usually suppressed to produce the antibodies. So depending upon the antigen, either the B cell get proliferated and differentiated to produce the antibodies, or they are completely suppressed. IgE antibody is the one that is involved in the hypersensitivity reaction. So it has both beneficial and harmful effect. In a normal person, this IgE helps to protect against the parasitic infections, whereas in atopic individual, this IgE is the one that produces the hypersensitivity or allergic reaction. So it was Ishikawa who has given us the concept of this IgE as a reagent antibody, atopic reagent antibody in atopic individual. It acts as to induce a hypersensitivity reaction in atopic individual. It has a molecular weight of about 190,000 Daltons, and half-life is only for 2 days, and it is a heat labile, and it has affinity for mast cells. So mast cells are the one that when the IgE binds, it degranulates to release a pharmacological active agent that brings about the elimination of the pathogen. But in atopic individual, it causes allergic reaction. So it was Pronitz and Kushner who reaction called as the hypersensitivity reaction help us to know whether the person is an atopic individual or a non-atopic individual. And this IgE antibody is mostly extravascular in nature, and it can be seen as in traces in a normal serum in a normal individual. But there is an elevation of this IgE serum in the people who are atopic or easily get allergic reaction due to hypersensitivity, and mostly seen in atopic individuals. And here these cells are distributed along the respiratory and intestinal tract, and when they go and interact with the mast cells of this respiratory and intestinal tract, they bring their clinical effect in the respiratory region or the intestinal region. So in the respiratory region, they causes hay fever, eczema, etc., whereas intestinal region, they can lead to food poisoning, and they are the one that are involved in anaphylactic hypersensitivity reaction. And they, in a normal person, they are the one that protects against the infection and helminthic infections. So depending upon when you summarize all the antibodies function, you can say that IgG antibody is the one that protects the person from the infection in the body fluids, IgA in the body surfaces because it is a secretory and can transport to the luminal surface, and IgM protects the person from the infection from the bloodstreams, and IgD helps in the recognition to activate or suppress the B cells to produce or not to produce the antibodies, and Ig is the one that is involved in hypersensitivity reaction. So the last part of the antibody molecule are the immunoglobulin specificity or variants in the antibodies. This is of biological importance when we are introducing one antibody from one species to the other species, how they develop an immune response towards this particular antibody. So it is of biological importance for us to know the different variants of the antibody molecule. So you can group them into 3 types. They are called as idiotypes, isotypes and allotypes. So based upon the differences in the amino acid that are present in different portion of the antibody, you can classify them into 3 important types: idiotypes, isotypes and allotypes. So idiotypes are the antigenic determinants that are usually seen in the hypervariable regions of the antibody molecule. So that is in the paratope, which interacts with the antigenic molecule. So depending upon these differences in the amino acid, it helps us to know with which antigen it is going to interact with. So idiotypes are antigenic determinants, or you can say that they are determinants which are present on the paratope or hypervariable regions of the antibody molecule. Now coming to the isotypes, as already discussed, there are 5 isotypes of antibody molecule depending upon they are structurally and genetically distinct. So based upon the different constant region, you can classify the antibodies into 5 major types. So it is the isotype type that helps us to classify the antibody into 5 major types. So they are called as phenotypic variants in the constant region of the antibody molecules of both heavy chain as well as light chain. So in the heavy chain, you based upon this isotypic determinants, you can classify them into IgG, IgA, IgD, IgM and IgG. Whereas in the light chain, you can classify them either into a kappa or the lambda type. So that is called as isotypic determinant. Now, ultimately coming to the allotypic determinant, it is nothing but the minor amino acid variations that usually helps to it is genetically determine differences in the antibodies between the people. So when we introduce this type of antibody with a different type of allotype, they usually produce immunological response towards this. So allotypes are nothing but the minor variations in the constant region of an antibody molecule. So all these variants help us to know whether they can be immunogenic in a particular species or in a different species of an organism. So this is all about the antibodies, which is the major effectors of the humoral immune branch. Reacts with the antigen, specifically depending upon the antigen, that particular type of antibody will be produced.